Today I am going to continue the previous talk where I will be discussing how to properly use cBioPortal and TCGA databases for different cancer types. It includes the introduction as well as the proper data exploration for specific types of cancers. So here, this is the previous slide where I had mentioned cBioPortal or TCGA as the major source of cancer data sets. And in general, if you would like to know about cBioPortal for cancer genomics, the website is there, cbioportal.org. Key features: it offers interactive exploration of genomic data across over 200 cancer studies. It includes data from TCGA, AACR, Project GenAI, and other major initiatives. Supports queries by gene, cancer types, mutation, copy number variation, and clinical data. Covers a wide range of cancers, including breast cancer, lung cancer, prostate cancer, skin cancer, brain cancer, etc. Pediatric cancers and rare cancers also. Tools for visualization, co-expression analysis, survival analysis, mutational mapping are also available. You can start by selecting a study or cancer type and then explore mutations, copy number alterations, mRNA expression, and the clinical outcomes. So, the key features, the comprehensive projects of 33 cancer types. Data includes genomic, epigenomic, transcriptomics, and proteomic profiles. It is accessible via the GTC portal. Now I am talking about TCGA, and it offers data download, web-based analysis, visualization. It supports integration with platforms like UCSC, Jena, Fire Browser, and cBioPortal. So, key features. Like for cBioPortal, I have mentioned already these things. Now you can understand the details here and then go for proper understanding what is your goal, what is the goal for your design project. Mutational mapping, co-expression analysis, survival analysis. Do you want to go for genomic, epigenomic, transcriptomics, and proteomics profiles? Now, the next part is like this is just sample presentation, how you can go for basic analysis. For example, here it is mutational frequency plot for selected genes in breast cancer. For example, TP53, PI3CA, PIK3CA, BRCA1, BRCA2, and PTEN. And you can see how it looks like in terms of plots, bar plot. And now, this is the pathway enrichment analysis. Of course, the plotting Style could be changed depending on how do you like and how best could be presented. Now, cBioPortal will be basically giving you mutational mapping, co-expression, survival analysis, genomic data visualization, transcriptomic data visualization, proteomics data visualization. And before you start, I'm basically showing the screenshots how you can proceed in cBioPortal to utilize TCGA database. For example, here I have selected a ML in myeloid, and here, if you can see, I have clicked the biggest data size data set. That is 2,330 samples are present in this data set. And then you have two options, either go for query by genes or explore selected studies. Most frequent analysis is like query by genes. And if I'm showing both of them, but here let's start from query by genes. Here, for example, I selected query by genes, and then my interest is to understand mutations, putative copy number alterations. Then I pasted the genes of my interest, which is in this box, enter genes. And all the genes symbols are valid. So this is also you need to check. And then click on submit query, and it will look like the outcome results will look like for example, cEBPA shows 7% mutation in case of AML, PTPN 11 shows 5% mutation, and RAS shows 13% mutation. So this is how you can go for it. And then you can also, you have the options, different options. For example, this is oncoprint, first option, but you have many more options, cancer type summary, mutual exclusivity, plots, mutations, co-expression, comparison, survival, pathways, and also downloads. And here you can see, in case of mutual exclusivity, how it looks like. The genes and the respective genes, A and B, are representing the respective genes, how they are dependent on each other, and then you can see the p-values, q-values, and tendency, and here it appears co-occurrence tendency. Then you can also, in this case, select what kind of like exclusivity you want to see in, then go for plots, and in plot you could go for different types of plots, and these are the examples. You can change the x-axis, y-axis depending on what kind of data plots you want. And then you can go for individual genes also, and click on it. You will see the mutations and its location, which is here in the figure, and then mutation type, whether it is missense, truncating, inframe, splice, fusion, what is happening. And then you can see more details about each mutations, whether it is insertion, deletion, and what is the allele frequency, many more things you can see here for each and every gene. Now you can go for survival analysis. In survival analysis, you can go for KM plot, and then you can also select the overall p-values for survival, or you can go for individual impact for each gene, and then go for each gene one by one also. Next part is, after survival, you can also go for further detail, the clinical relevance of these genes and the samples, what you have it here. So for example, here you have, let's say, TMB, non-synonymous, then what kind of test has been performed, and what is the p-value, what is the q-value, then patient's vital status, race, ethnicity, patient details, so many more things you can go for it. And further, you go for example, you select different genes and go for their further details. clinical relevance in terms and the genomic alterations. Then next part is mRNA, which you can see here as a volcano plot for all the genes which are expressed here. Then next part is the details of all the genes, how high expression, low expression, what is the log2 values, log2 ratios, p values, everything, and then cytoband also, which may not be. And the last part in cBioPortal will be to look for the genes they are mutated and what is their phenotypic relevance, followed by their presence in biological pathways. And here, this is the highest significance pathway in this selected genes, and its impact, overall impact, goes to proliferation, cell survival, translation, which you can see here, these three biological functions. But the genes which are most affected are KIT, FLT3, then PTPN11, KRAS, NRAS. These are the main genes. Now, next part is the further details in terms of mRNA expressions. More details you can see. And finally, you can go for the next part where you can explore the selected studies. Here you don't need to include the genes from your list, but explore overall changes in mutation, copy number alteration, gene expression, or structural variants, everything you can. So what you can explore, this is the map. For example, here you can see mutated genes, all the genes which are mutated, and copy number alterations, and many more things. Then also here you have the diagnosis ages, that means the patient's details, patient's like gender, ethnicity, race, everything you can explore for the selected studies. And if you want to explore the details in terms of clinical perspective, then you can click on clinical data. Then you could see such kind of details like whether it has been diagnosed, and if it has been diagnosed, then how it looked like. All the details, clinical details you can see here. And then last part is to go for plots. Here, this is